Hi, so in this week's module, we learn about transcription and translation. Transcription, it happens when the gene of DNA is copied into mRNA. First, we're going to have a molecule of DNA. And the promoter, we're going to need a promoter to bind where the gene is, so enzymes are able to bind to the gene. So first we have RNA polymerase is going to be able to open the DNA. And then RNA polymerase is going to add RNA nucleotides building the mRNA. So here we see the building of mRNA. So here is only the gene that is going to be coded, that'll be encoded. So here we have the promoter before, so now RNA polymerase knows where to bind and start the transcription. The termination occurs and end of the gene mRNA is released. So here are the major differences between transcription in prokaryotes and eukaryotes, because in prokaryotes, translation begins immediately. Because there are no nucleus, ribosomes are able to bind and begin the translation. Different in eukaryotes. Since they are inside the nucleus, transcription happens inside the nucleus. So translation to happens this mRNA initial leave. But first, what we have just transcribed is a pre-mRNA that has introns and exons, made of introns and exons. So the introns are not going to be coded for the final protein, but only the exons. And in order to remove these introns, we pass through a process called splicing that removes the introns. And what is going to result in mRNA made of exons. And also it's added a 5' cap and a poly A tail. The 5' cap is made of methylguanine nucleotides, and the poly A tail is made only of adenine nucleotides, and they're added to improve the stability, and they can be reused. Another difference between bacteria and eukaryote is that bacteria are monocystronic or can be polycystronic. Polycystronic means that the mRNA can encode for many different polypeptides. In eukaryotes, they are monocystronic. mRNA is going to only encode for one polypeptide. In transcription, there are also other differences, including that in eukaryotes, since it happens in the nucleus, they have 5' cap, 3 poly A tail, and a splicing happening. Here we have the other process that it's called translation. That's when the codon it's going to be able to bind and encode for amino acid. So translation is also known as the protein synthesis. Amino acids are the building block of proteins, so they are made in different sites in the ribosome. So first, the ribosomes have 3 different sites. They're A, P, and E. In the site P, it brings the tRNA. tRNA reads the mRNA codon and brings the correct amino acid. So the anticodon that the tRNA has, it is complementary to the mRNA codon. Each codon encodes for a different type of amino acid. First, and at the codon P, it is this tRNA and the anticodon recognize, and they're complementary, and they're together form the first. They carry the amino acid and they form methionine, and then in the site in the A site, we're gonna have the tRNA binding to the codon CCC, which is going to result in proline. And this proline and this methionine, they're going to bond, forming a polypeptide bond. And then this protein, this ribosome is going to shift, and then they're going to have another tRNA binding to another codon and adding another amino acid. And this process is going to be done over and over until it reaches a stop codon. And the stop codon is going to release a factor. This is going to cause the release of the polypeptide and terminate in translation. So in the end of the translation, we're going to have a polypeptide as a result. So in this process, the ribosome is able to facilitate the formation of peptide bond, and also the A side that I haven't mentioned before is where the tRNA is going to exit. There are also other differences in the translation process between bacteria and eukaryotes. So first, they have 70S ribosomes, and different from eukaryotes, they have 80S ribosomes. So the first amino acid that is encoded in eukaryotes is called methionine, and it's different from bacteria that's called formylmethionine. And another difference between both is that translation cannot happen fast, so it needs to pass through this process of splicing, where bacteria can happen simultaneously, transcription and translation.